Good morning, everybody. So today we are very happy to have Elise Robinson. Elise is a assistant professor at the Center for Genomic Medicine and Department of Psychiatry at MGH. She is also an institute member of the Broad. She is also affiliated faculty with the Analytic and Translational Genetics Unit at MGH. The Robinson Lab uses techniques from statistical genetics and epidemiology to study how common and rare genetic risk factors for severe neuropsychiatric disorders may differ, and develops methodological approaches for examining these questions in large samples. Elise received her doctorate degree in psychiatric epidemiology from the Harvard School of Public Health and also completed her postdoctoral training here at the Broad at MGH. So thank you for being here. And I think if anyone has questions, just raise your hand and ask, come to the mic, and we look forward to the discussion.
All right. Let's talk about GWAS. There's a lot one could talk about. I'm going to focus mostly on conceptual overviews and where we've come, current uses, and points of epidemiologic caution. So please ask questions throughout if you would like, including those of you who are on Zoom, who I cannot see. Starting with the history. So we do GWAS to study complex traits being human phenotypic outcomes in which there are many hundreds of relevant genes, or for which there are many relevant genes. And one could, through complex trait genetics, study common variants, which are those traditionally referred to at least as having a frequency in the population in excess of 1%, or rare variants, which range in frequency from private to just less than 1%. Today, we're going to talk about common variants that are present in more than 1% of the population, and are typically thought to explain the majority of variants in human traits. They're fairly stable between human populations, at least as compared to rare variants, and associated with a large fraction of phenotypic variants in human traits, and a vast variety of traits. A lot of my examples are going to lean towards psychiatry because I'm in the Stanley Center and that's what we do. But this is true for everything from height to blood pressure to schizophrenia, which we'll talk about a bunch, a vast array of human traits. So having believed for quite a long time that common complex variation was going to influence a huge amount of human trait variability, people began to pursue GWAS when technology afforded the ability to do so at reasonable cost. And I have included this lovely video from NHGRI EBI that tracks GWAS findings over time. So this shows the progression. How do I make it play? Oh, no, I found it. We're good. Progression in GWAS hits. So the number of specific loci associated via common variation to any human trait and submitted to this repository over the last 20 or so years. So let's start with January of 2006. And now we have over 1 million GWAS hits that have been cataloged in total. So what are we going to do with them? Nope. There we go. Let's look at that growth across one phenotype, so specifically schizophrenia. Schizophrenia has long been thought to run in families. In fact, when people began writing about complex trait genetics, it was included amongst the early traits that people thought were highly heritable. And twin studies, which we'll discuss the kind of dark art of a little later, have also long suggested that it's highly heritable, and it made sense to pursue it in GWAS. But the first GWAS efforts in schizophrenia were not successful. This is a genome-wide association study in schizophrenia with 2,600 cases. And it's hard to know how much about GWAS people in this room already know, but late and brief. The idea is you scan millions of variants that are common in the human genome for association to your outcome. In this case, comparing frequency of common variants, again, millions of them, between individuals diagnosed with schizophrenia and not diagnosed with schizophrenia. Because across those millions of tests, you do something to the equivalent of 1 million independent tests, we correct the significance level down to 5 times 10 to the minus 8. And in this first effort in schizophrenia, there was no common variant amongst those scanned that reached that level of significance. So there was no genome-wide significant association with schizophrenia in the first GWAS. Would anyone like to guess why? I don't know if someone in here knows. You totally know. It's because the sample size was small. So the expectations one can have for a genetic study of common variation is dependent on the extent to which your outcome is associated with fecundity, or specifically a reduction thereof. So schizophrenia, for example, is associated with about a 75% reduction in the number of children one has if they are male, about 50% if they are female. This is a very strong impact on the number of children one has. And what that does is institute a mathematical limit on the extent to which a common variant can increase risk. Because anything that creates a large amount of risk for an outcome that reduces the number of children one has on average, especially quite substantially, cannot become common in the population. And it can only survive even at lower frequency for a few generations. And at this level of fecundity impact, we don't expect common variants associated with schizophrenia to have an odds ratio above 1.1. So what's happening here is that if you're looking for an odds ratio of 1.1 or lower and correcting your p-value down to 5 times 10 to the minus 8, 2,600 people simply isn't enough to identify a small association with that level of statistical significance. So here we go. We have 2009, 2011, 5 GWAS hits with 9,000 cases. 2014, 37,000 cases, 108 loci at the World Congress of Psychiatric Genetics. Stefan Ripka got a standing ovation. Everyone was so excited. This was the first time something like this had happened in psychiatric genetics. And within that 2014 data set, one could see indeed evidence of these selection effects and what was necessary from a power perspective. So this was called by Ben, Neal and Mark Daly, the power smile. I should add Ben, Neal, and Mark Daly, from who I took pretty much all of the slides up until this point, and then I will continue to acknowledge those that I've collected slides from for this talk over the last vaguely 12 years. Okay, so at the lowest frequencies, you're allowed a little bit more bite in the odds ratio, but the more common we get, the more it has to stay south of that limit. And the reason they're clustered here at 2014 is up near that expected limit is because that is what power is allowing them to find, even at the, whatever I said, 37,000 cases. If you want to drop down here, you keep increasing the sample size, and then you'll get more hits. You'll be powered to get genome-wide association of variants that have even smaller odds ratios associated with them. And indeed, that is where they got to in 2022, 76,000 cases, 287 distinct loci. Okay. And this isn't true for all traits. So there are traits in which the average age of onset is past the human reproductive period, especially the human reproductive period as it was typically defined many thousands of years ago. And so, for example, breast cancer and macular degeneration have large impact common risk factors, but for traits that are early onset fecundity impacting or exceptionally genetically complex, typically these odds ratios are quite small. So how much do common variants matter? And this is typically, have you guys heard about heritability? Where are we starting? Yes, generally yes. Okay, heritability is the proportion of the variation in a trait population-wide that is explained by genetics. When thinking about GWAS, we typically are thinking about narrow sense heritability, and specifically the proportion of trait variants attributable to additive genetic effects, meaning this on top of this on top of this, not this effect modified by this other one. And you can estimate heritability lots of different ways. Historically, people did it by comparing monozygotic and dizygotic twins, the idea being that monozygotic twins were twice as similar genetically as dizygotic, but shared environments and many other things. Involves a lot of modeling assumptions. It typically produces estimates of heritability in excess of other family-based or SNP-based methods. But those also involve a lot of assumptions and modeling choices. Relatedness disequilibrium regression, people now often commonly use comparison of direct family members, which produces on average lower, for example, 55% down from 80 estimates of heritability. And then you can also use directly measured SNP data. So the results of a GWAS, there are technologies like LDSC or GCTA that estimate the amount of variation in an outcome that is attributable to the additive effects of basically SNPs on the chip. So a lot of you have probably heard about missing heritability and how much do we care that the SNP heritability is typically half or less than half of the twin heritability. And 10 years ago, I was very interested in this. And now I just don't care at all. Because I think if the goal of these activities was just to try to discover as much common variation needed to fill what the twin studies said was important, then we'd have a problem. But as I'm going to talk about on the next slide, that actually has nothing to do with why I'm interested in common variation. And no matter what, doesn't matter if height is 80% or 50% induced by common complex variation, common complex variation remains by far the greatest influence on height. And so why wouldn't one include the greatest genetic influence on a complex trait in one's explorations of its underlying biology? So anyway, 80, 50, I'm fine with it. And that's even more stark for a lot of behavioral traits. You know, the twin average for a lot of behaviorally based traits is probably about 50 or 60, and then SNPs is way lower for a variety of reasons, both because you're going to get the general height drop, but then also likely because of the manner in which heterogeneity and measurement issues disproportionately impact SNP versus twin data. But same goes, I don't really care. If any of you do care, please feel free to make an argument to that effect, and I'm happy to hear it and maybe change my mind again, but probably not. Okay, so why study common variation if we aren't super worried about how much it explains in total, at least relative to twins? I am interested in using it to understand biology. So for some traits, GWAS standing alone has provided clear insights into biology that have been immediately translated. People typically use TYK2 for psoriasis as an example, where people found that variants that increase the expression of this gene increase risk for psoriasis, and then variants that reduce expression of this gene are found in healthy people, which makes it a great drug target. And now there are treatments based on that. So for outcomes like those in psychiatry, where common variation has not and almost certainly will not flag for us some critical core process thanks to one SNP. I do believe and am interested in developing methods to understand the biological impact of aggregate common variation, but for, I think, most common traits, single SNPs are difficult to parse, particularly given the volume of the SNPs associated and the fact that the majority of them have regulatory functions that are difficult to explore, which is not going to be the focus of this talk, but it is harder to understand the biological impact of a GWAS-associated variant than a coding variant in a gene. And some of the reasons that's true are most GWAS hits lie outside of genes. And in order to figure out what they're doing, you first have to figure out the causal SNP. A GWAS finding typically is referred to as a tag SNP. And since common variants move in packs or LD blocks, the variant that comes out of the GWAS is often just the most common one in the LD block, or it has various other statistical but not necessarily biologically interesting properties that render it the one that came out of the block. You have to find the causal SNP, which is a statistical process necessitating a lot of samples and often a lot of patients. Then you have to figure out what that SNP is doing. And for most of them, it's not, reducing the function of gene X like you get from our favorite coding variants through exome sequencing. It's engaging in a regulatory process that's highly variable across the genome and really typically existing within a space of biology that we're only beginning to understand. So, while pursuing GWAS hits functionally is hard, and it's certainly harder than, for example, pursuing the canonical approach of knock out gene X in a mouse to understand what this loss of function variant in a person might be doing. Knocking out gene X in a mouse isn't easy either. And particularly as we enter an era in which, for example, for schizophrenia, the rare variant association studies are producing now quite long lists. For autism, those lists have been in the hundreds for neurodevelopmental disorders, I should say, those lists have been in the hundreds for a while. For autism, they're moving into the hundreds now. What is our plan? Of all those genes associated with complex trait outcomes now through rare variant sequencing, specifically genes, how are we going to choose which ones to parse first? Where are we going to place our bets when it comes to pursuing the biology implicated by deleterious variants in those genes? If you're given a list of 200 genes from exome sequencing, are we just going to throw them over the fence at the functional biologists and say, please give us the biology of all 200 of these and figure out the stuff that's the same and not the same? I think multiple classes of variation, studying multiple classes of variation can really help in that prioritization activity. So if, for example, you have 250 genes coming out of the next autism effort, and you're trying to think, which one should we study, which one should we pour our millions of dollars into pursuing the functional implications of first? Why not start with genes that have been nominated through multiple classes of variation as relevant? And one of the nice things about common variation is is common, and it provides increased confidence that the processes that you're going to be unpacking with these focused gene studies might be more relevant to more people, might be a more globally relevant process. For some complex traits, definitely not all, I think we also need to remain agnostic to the possibility that different classes of variation may implicate different types of biology. There's this really nice paper by Dan Weiner, Ajay Nadig, and Luke O'Connor that was published a couple years ago that suggested for a lot of complex traits, rare and common variant influences both implicate similar processes and pathways, and rare and common influences, for example, on schizophrenia, have similar associations to other phenotypes, meaning genetic correlation, which I'll introduce very briefly later, derived from rare variants associated with schizophrenia look a lot like the genetic correlations associated from or derived from the common. There's some traits where we can't be as steady about that. Two of them come to mind for me, given the research I do, are autism and age of walking. So the rare variants on average associated with autism spectrum disorder have a negative association with cognition in the general population. The common variants associated with autism, I co-chair the Autism GWAS Consortium, are positively associated with IQ, both in the general population and in people with a diagnosis of autism. Age of walking is actually the same. Delayed walking is associated with, unsurprisingly, a lot of rare genetic variants that are associated with syndromic developmental disorders that impact global learning, motor delay, hence the term global developmental delay, but that includes walking. On the other hand, later walking is also associated with common variant in predictors of higher IQ and greater educational attainment in the general population. So while we have fully characterized neither of those things well enough to really look at the processes and pathways question biologically, we already know that the phenotypic associations are not just one is a muted version of the other, but they're opposite direction. And that doesn't provide a lot of confidence that we should just assume the biology spoken to by the common and rare variants will be the same. So agnosticism is where I land.
We have a quick question online.
Yeah.
Can you clarify for these diseases and traits that have hundreds of known variants, for any one individual, do they just have one of these variants or all of them, or how does that distribute across the individual?
Great question. So it matters a lot about whether we're talking about common variants or rare variants. Rare variants that have, for example, a large impact for brain-based traits like the ones I'm talking about, people typically have one if they have any. For common variants, we all have some degree of aggregate, many thousands of variants, many hundreds of genes, genetic risk for schizophrenia because it's some distribution in the population, which is what we get with the things we call polygenic risk scores. We all have some aggregate position on the genetic distribution for height. For any complex trait, we all live somewhere on that distribution. Let's get to actually PRS, and that might help with the question. So a polygenic risk score is, at its core, a very algorithmically simple thing. So you do your GWAS, you've got your many millions of common variants associated with some trait outcome. Most of those associations are not statistically significant in and of themselves. The idea of a polygenic risk score is you just go down your list of associated SNPs and you multiply the number of risk alleles you have, 0, 1, or 2, by the odds ratio associated with that allele. And then you add that up all the way across the genome. There are many different forms and flavors of polygenic risk score construction. Some that are just the thing I just said, which you could do in an Excel spreadsheet, and some that are decidedly more black boxy. The more modern ones do often provide a little bit more predictive ability or have some nice features like better flexibility across different ancestral populations of humans. However, by far, the thing that best predicts how well your polygenic risk score is going to predict anything is the sample size of the GWAS. So for any complex trait, you end up with a population-wide distribution of polygenic risk, just a normal distribution. And For case control studies, you can imagine patients, non-patients, even in the largest and most beautiful of GWAS, you're not going to get distributions that are non-overlapping for a truly complex trait. What you will get is a little shift. So just people at the top of the distribution are probably more likely to be cases. People at the bottom of the distribution are more likely to be controls. And that's what this is showing. The distribution's just shifted. Any questions about that? Did that answer the question on Zoom? Okay. I want to pause there just in case there are any other questions, because now we're going to talk about the epidemiologic confusions of GWAS and what is in my score.
I have a quick question. So you had that curve and you're showing it like 50% allele frequency. If you had even more people, you could get odds ratios like very close to 1 that are still significant. Do you think it's worth getting more samples to find those? Like how biologically relevant are those and how much more will you learn about the pathways by finding them?
Question, and one that we wrestle with a lot. People have different answers, and I think a lot of it relates to what they're hoping to get out of a GWAS biologically and quantitatively. It also relates a lot to just belief systems around utility of different forms of genetic variation. I can say for me personally, the reason you would want more than this is A, if you're only getting variants that exist at kind of the minimum or at the top of that odds ratio threshold to be detectable, there's no way you're getting a particularly strong statistical characterization of those effect sizes. And it could just be a lot better with more people. And you can realistically do a lot more with a gene list or with a SNP list that is in the 200s than it is in the 100s in terms of lining up rare and common variant influences. Other people say we want to keep going until we find redundant biology. For some traits like height, they've reached a point of saturation where they have found all of the associated variants. They have very tight estimation of, or they have tight estimation of the effect sizes of those variants. In schizophrenia and autism, I don't think we're ever going to get there, but we have a lot farther to go in improving our estimated associations in a way that could uncover novel biology. Okay.
Going back to that smile figure, I don't understand the right side of it.
Why it's smiley, not just like a smirk?
Yeah.
Yeah. So it's just because the risk allele is the less frequent one. So like here, yeah. So it's just a flip in the relevant allele. I agree though. Like it could have just been a, yeah. Okay. What is in a PRS? The answer is basically just your trait plus. I think, and this is changing a bit, but historically people pursued genetics using epidemiologic samples without an eye towards the epidemiology of those samples. And something that is critically important to remember when working with GWAS results or GWAS data is that in a lot of ways, we are bound by all the complexities of observational epidemiology more generally. So when you do, for example, a GWAS of depression, you are doing a GWAS of depression. You're also doing a GWAS of every single thing that is more common in your cases than in your controls. And think how many things that are deeply complicated create risk for depression and will be showing up more commonly in cases and controls. That means your GWAS is also containing heart disease, chronic pain, job loss, a huge amount of additional factors that then end up populating the patterns, the genotype to phenotype patterns that arrive through use of that score. So when I wrote my first R01, the idea was that we would use polygenic risk for autism and a bunch of other psychiatric disorders, and rare variant risk as well. But we'd basically try to class genetic risk factors for psychiatric disease based on their distribution of phenotypic effects in a manner that could inform us about their effectively biological proximity. I wrote that in 2015-ish. I now think that's totally wrong. So I have now kind of become convinced that using polygenic risk scores to understand phenotypes in the human population is no less fraught than any other form of observational epidemiology. And one has to put on a very tightly attached thinking cap in order to interpret the distribution of associations you see from it. So here, this is from a version of a UK Biobank analysis that Caitlin Carey, Raymond Walters, Robbie Weedo, Rebecca Shaffey, lots of people in the Stanley Center worked on. We conducted a phenotypic factor analysis of hundreds of outcomes in the UK Biobank dataset, which has tens of thousands of them, if not more. And this is an early version of the factors that came out on the x-axis. So for example, the heart attack factor has a strong genetic correlation with coronary artery disease or genetic risk, polygenic risk for coronary artery disease, which it really should. If you have higher polygenic risk for coronary artery disease, you should have a higher score on the history of heart attack factor. However, heart attack is as associated with polygenic risk for major depression as it is with polygenic risk for heart attack. And you'll notice that ADHD is up there as well. And that more broadly, the distribution of associations to the ADHD polygenic score looks a lot like the distribution of associations to the depression polygene score, but not so much like autism, bipolar disorder, or schizophrenia, or IBD. A little bit like coronary heart disease. Looks more like those than it does the other psychiatric traits. Any thoughts as to why? What might a diagnosis of ADHD in adults and a diagnosis of depression in adults on average come with that these other three psychiatric disorders? Well, actually, I shouldn't say the diagnosis. It's actually a hard question to pose. What do ADHD and MDD more likely have in their epidemiologic package than the other three? The answer is basically lower socioeconomic status at a family level. Don't think of it, particularly for schizophrenia, don't think about it from the perspective of the diagnosis of the adults. Think about it from, for example, educational background of the parents. And this is obviously an average thing, but in the US, kids are more likely to be diagnosed with ADHD if they come from a less privileged socioeconomic background. And depression is similarly more likely to arise in people who have more strenuous life circumstances over the course of their lives. And What that means is the GWAS is picking up on those average differences as well as the ones that are directly related to underlying risk for ADHD and depression. And those things populate the suite of phenotypic associations to those scores. Does that make sense? I saw some confused faces. To be clear, that doesn't mean that lots of people with, for example, a diagnosis of ADHD don't come from families with very high educational backgrounds, don't themselves have high educational backgrounds, or are not like overrepresented in people who work in this building, because they are. But on average across the population. Go ahead.
Oh, yeah. Thank you. I have a quick question. I'm just wondering about the heritability of those psychiatric diseases are similar or different?
Schizophrenia and bipolar are the highest. Autism is typically estimated in the middle, along with ADHD, which is very high, and MDD is a bit lower. But
the kind of difference of heritability could affect the different genetic correlation distribution or that?
the genetic correlation, at least is estimated here, is done in a manner that's meant to be independent of both sample size and heritability.
Thank you.
I have one question about that socioeconomic factor, because like what I don't understand is why this should be so much stronger than as a reason to have that correlation than like any other factor. Like why would you say, okay, the socioeconomic status is the reason why they're correlated with each other and not any other trait they have in common?
What do you mean by any other trait they have
Like
in common?
like we see like 10 things that are like highly correlated probably, and then why would you say that like the lower income is the thing that is like driving the correlation among all the other traits?
Fair. There probably are many things that could be related to it. However, for example, educational attainment is heritable. And in places where access to healthcare and resources is dependent on a family's financial background to any degree, you see these sorts of patterns where the socioeconomically linked traits travel together. Does that make sense?
Yeah, it makes sense.
Thank you. I also wanna ask one question because I don't know. You said it's in the analysis was conducted in the UK Biobank. Could it be possible that some of the I don't know disorders or diseases they are just highly comorbid, have a high I don't know comorbid rating in the data set?
So these polygenic scores are common variant GWAS results for ADHD, ASD, bipolar disorder, major depressive disorder, and schizophrenia. Weren't generated from GWAS and UK Biobank. They're generated from the Psychiatric Genomics Consortium that takes different, many thousands of people with these diagnoses from all over the world. And then those GWAS results were used to create I don't want to conflate polygenic risk scores analysis with genetic correlation analysis, which was done here, but it's a little hard to explain without going into details with one or the other. The genetic risk for the psychiatric diagnoses was not generated using data from individuals from UK Biobank. They're independent. Does that answer your question? Because this also isn't specific to UK Biobank. You see this in these sorts of patterns in the US. You are quite likely to see them less in economies with flatter income structures. So I think the point is you have to be very cautious of the totality of what's in your GWAS. And those add-ons, those pluses, can vary a huge amount between contexts. Another thing that can vary a lot between contexts is diagnostic tradition. So genetic correlation, which in common variant space just means the degree to which two complex traits share their genetic influences. A genetic correlation of 1 means that 2 complex traits share on average all of their genetic influences. -1, all of them, but in the opposite direction. 0, none, 0.4, 40%, et cetera, et cetera. So if you estimate a genetic correlation between common variant influences on autism and ADHD in Denmark, it's about 0.4. So using Danish data, it's about 40% sharing. Using US data, it's about 0.05 currently, which is very different. So 5% sharing versus 40% sharing, which is quite likely a function of different diagnostic traditions. And so what you end up in your GWAS and how you end up interpreting it and studying biology could be a mix of, fairly stable features of a trait and then fairly complex or context-dependent features of a trait. Here's another example from UK Biobank. This is a paper from Andrea Gana's group, which I really love. 8 minutes? Yeah. So we'll just get to the end of this. In UK Biobank, Nicola Perazzu in Andrea's group did this very cool analysis. She did a GWAS of just sex, male versus female. And this is on the autosome. So the 22 chromosomes that are not different between men and women. There are no known autosomal differences on average in common variant space between men and women. So theoretically, if you go into UK Biobank and you just do a GWAS of sex, what should you find? Nothing. Nothing should come from it. So much came from it. There's a giant sample size. And this happened because you're not doing a GWAS of sex population-wide. You're doing a GWAS of being a woman in the UK Biobank versus being a man in the UK Biobank, and more specifically, being a woman who signed up to be in the UK Biobank versus a man who signed up to be in the UK Biobank. And one of the things they found, this was exceptionally statistically significant, even though a very tiny little difference, but it was exceptionally statistically significant because they have a massive sample size, was that men in the UK Biobank were more likely to carry a weight increasing allele in the FTO gene than women. Is that true population-wide? No. So imagine a population in which you have a similar distribution of BMI or of weight between men and women. You end up with GWAS hits related to weight in a GWAS of men and women in UK Biobank if, in lieu of a representative proportion of men and women joining, you get this, where there's a difference in the probability with which men and women will sign up for the study, and that difference is associated with BMI. And one of the nice things they found is that indeed you can do things like this with GWAS data to interrogate participation bias across different studies. Participation bias is historically very difficult to understand just because you can't analyze people who didn't sign up. But using things like this, you can use genetic data, common variant GWAS data as an instrument to understand patterns like this. And across a variety of data sets, they found that you see evidence for heritability of sex from the autosome where there shouldn't be any in studies where you had to actively sign up. In this case, it was 23andMe and UK Biobank. You don't see it in studies where you do not have to actively sign up. You passively are engaged, and that's more like the European biobanks. And we have to be cautious. So all of these data sets come with their positives and negatives, right? So in the UK Biobank or case control studies where you have to sign up as typically a case, you have these participation effects that do enter into your consideration of genetic influences, particularly genetic influences that are subtle. And then if you were to focus on, for example, the biobank related resources, you have cleaner population representation, but you're often left with only what's in those data sets because people aren't being contacted to provide additional information. So to give an iPsych example, I co-chair the Psychiatric Genomics Consortium Autism Group with Anders Borglum, who's a member of the Danish iPsych Consortium. And a lot of our aggregate data comes from this iPsych resource, which is a Danish population registry. And so we have a fairly representative, if not a census, of autism diagnoses in the country, but we don't have extra information on them. So it becomes really hard to figure out, for example, why exactly the genetic correlation with ADHD is really high in Denmark and not so much in the US. It's very hard to figure out, for example, why if you take the Danish data and the US data, the autism in Denmark and autism in the US have a fairly low genetic correlation for being the same trait. We can't dive in because we don't have additional phenotypic information because the participation wasn't specifically requested. And I think we're just going to stop there because there's another section that we don't have time for.
If you have any questions, please come up to the two microphones. Thank you.
Hi, thanks for a beautiful, clear talk. I have two questions. The first, I think, relates to some of the other questions that have been posed. I'm interested in what it means to you when you say that you're aiming to understand biology.
I think it depends a lot on the trait. You mean just me as me? What do I want to know? Okay. I'm an autism researcher. I am interested in understanding the biology of experiences common to the autism community, common but highly variable, and specifically understanding it to a degree that will eventually allow us to help people who want to be helped in the ways that they would like to be helped. I think there are lots of simple answers. So the psoriasis example was, what causes psoriasis so I can make a drug for psoriasis, right? I think separately, I'm also interested in understanding biology, at this point, the regulatory impact of common coding variation, because in a trait agnostic way, that's something about which we're still quite unclear. And one of the, I think probably my greatest statistical interest at the moment is to develop statistical methods to better interrogate the functional impact of aggregate common variation.
Yeah, I was interested in how much the word mechanism would come up in your response, and like the distinction between probing correlations versus something else and what that meant to you. But I was also really interested that you framed your whole talk as kind of like a defense of the study of common variants. And I wonder if you could give us a flavor for people who don't know the field so well. Do you feel like the study of common variants is under attack in any way, or just interested in that framing?
I made that framing, but perhaps it's because it's always under attack. Like it's never not been under attack. But then I think the longer you're in genetics, the more you realize that everyone is generally under attack at some point. You know, people just have very diverse opinions. And I think what might have come up as or might have come across as a defensiveness was more a questioning of why I still do it. And why I am interested, because I think that has evolved a lot over the last 10 years. And the early ideas of what GWAS would produce and how quickly it would be able to create change for patients haven't come to fruition, but I remain interested in the thing that it is.
Go ahead.
Hi.
I have a question about the autism and ADHD data sets that you work with. When you consider controls, what exactly do you count as a control? And we know that especially like women are undiagnosed. Do you think any of your controls are misdiagnosed or undiagnosed, or do you see any changes in your data sets as people are getting later in life diagnoses?
In fact, the 10 slides I didn't do were about that for autism.
Great.
So let's start with the ADHD one. By the way, I shouldn't have made a joke about how ADHD was, quote, overrepresented in this building. The point was I just didn't want anyone to think that I was suggesting that ADHD is not indeed present across the distribution and maybe quite common amongst statistical geneticists. So ADHD, to go back to your question, functions beautifully as a continuum, both etiologically and phenotypically, meaning the genetic influences on ADHD, the diagnosis, are very correlated with the genetic influences on ADHD as a trait that you can measure kind of quantitatively in people, typically through symptom counts. And so in that context, one can think about case control stat as much as one can think about kind of height, where if you do a GWAS of tall people, your controls are going to involve a lot of people who, are kind of tall, but the majority aren't. And so there are statistical pluses and minuses. A lot of the trait measures are perfectly correlated. You can probably access more diagnostic data than you can. There are reasons to approach it in multiple ways. Autism is so complicated, and it is because of that complexity that I will remain forever dedicated. So with the question about women, how much time do we have? A minute.
You could chat after.
Yeah, let's just chat after.
Okay.
But I think to go to the ADHD thing, if you jump from that description to autism does function not anywhere near as well as a phenotypic and etiologic continuum. And that's probably for a lot of reasons. But there's a lot more reason to maintain a case control framework, even though caseness continues to evolve and does continue to disproportionately affect certain demographic groups. But I was going to try to do it in the last 4 minutes, but that seems irresponsible.
So your controls are people that have been assessed or no, controls can also just be, we have the DNA, but we don't.
Yeah, and I think whenever one is doing a case control study, you have to entertain the possibility of certain misclassification of bias. And even if you were to say, in the autism case, let's say vaguely 2 to 3% in the US population currently meet criteria, receive a diagnosis. Of course, around that number, there's some wiggle in who could be diagnosed or who may eventually be diagnosed. But that number is never going to reach a point where the majority of your controls could be or will actually eventually become cases. So it's a statistically tolerable amount of potential misclassification bias that is conceptually voluminous.
Great, thank you.